Good morning, everyone. Welcome back to our session. Today we're going to talk about sequencing. So let's just make sure that everyone is here. Okay, please make sure that you have sound and you can hear everything. If you don't, maybe you have to reconnect. So welcome back, everyone, to this program. Today, we will talk about metagenomic data. So how is this data generated? What are the different types of generating this data? What are some terms and formats that you have to be aware of so that whenever we make our progress into the next steps of the analysis of this data, we'll be all on the same page about what kinds of data are there and what we can do with that kind of data. So really today it's going to be an introduction to next generation sequencing for metagenomic sequencing, whole genome, 16S ribosomal RNA, and amplicon data type. So there's a variety of different ways to produce this data. So to study microbial communities or microbiota, we will learn about these different methods of sequencing and how that impacts the selection of different downstream analysis and processing of the data. The good understanding of the way this data was prepared will actually help us navigate different types of research studies and read library preparation protocols that is typically going to be mentioned whenever you come across a data set. So remember that metagenomics refers to both a research technique and a research field. Specifically when we talk about metagenomics as an approach, we want to analyze microbial DNA from environmental communities. So we want to preserve the composition or the condition that a particular member of that community is in. Metagenomics analysis tools enable us to study population that consists of both culturable and typically unculturable, or a lot of unknown microbes. And that's really one of the challenges, is that when we speak about this community, it will have different levels of abundance for different types of species. Many of the individual members are going to be very poorly studied and unannotated. So there are a lot of challenges, only 1 to 2% of bacteria actually can be cultured in the lab. So learning about these other members of these communities is challenging, and we have to rely on similarity and try to infer from that similarity how might this particular member be different or similar to other members that we do know something about. But the ability to identify microbes, so determine which microbes are present and how they are related to each other without a priori knowledge of what the sample contains, is really opening new doors in disciplines like microbial ecology. When we look at the environment where microbes are abundant in both soil and water, virology, these communities contain not only bacteria, but also viruses, microbiology, environmental sciences, and biomedical research. And so we'll look today at a few examples of how sequencing data could be found and what types of projects we can find. There's a lot of them, so we'll see that. But before we go on, before we jump into the topic for today's session, I just briefly wanted to take a look at the progress of the group overall and kind of talk a little bit more about how to make the most out of this program for each one of you. So if you go to our program page, this is the program page. On this program page, you can scroll down and you can see here, this is the section that we're on. So this is the session. You can read a little bit more about this session. And then you can scroll down and you will see the activity. So all of your activity is going to be listed out here, and our goal with the tracking this activity is to help you overcome any kinds of challenges and kind of understand what is, the balance between theoretical and practical and maybe project progress that you have. So we're really looking at this regularly, trying to make sure that the outcomes are not going to be slowed down by any technical issues that come up. So the thing that you can also find on this program page, if you scroll up, is this progress tab. And so in this progress tab, what is displayed is the average number of points that you accumulate as a group. And we can also see some of the more active members that have been recently doing something and taking practical steps, running some code, or running pipelines, or completing some coursework. So this is informative for us, obviously. We can learn from this, but also I think that all of you can learn from that as well. The ideal way that we view the program overall is we want to help you to get to a reasonable number of points, which is kind of a proxy for us to understand that you've been actively participating in the program. So the average person earns anywhere between 5,000 and 10,000 points during a program. So what are these points given for? You can complete a course for 500 points, run a pipeline for 100 points, update your profile for 100 points, or have some group activity. Now, there's also going to be coding challenges, little coding blocks that you can complete or lessons that you can complete. Those also range between 50 and 250 points. So the average user generates about between 5,000 and 10,000 points during a program. Typically, if you're just doing the minimum, you're going to get around 5,000 points. If you are practicing all of the activities that we do together and doing things on your own, as well as working on an independent project, you should be well over 10,000 points by the end of the program. So looking at those points just kind of gives you a sense of where am I in my own progress, and where am I compared to some of the other participants in the program. For example, some of the profiles, just to kind of give you an example, Carmel is here with 750 points. You can see that she completed the introduction to bioinformatics. Mikhail, 25,000 points, 810, and that's because you can see that he's already completed some of the other programs previously. So that's how you can kind of start telling the difference between different people in the program. So people that are just starting to explore things, they're new, they're starting to learn. Some of the other people are going to be more and more advanced. Now, as the time goes on, you'll see that practicing some of the things independently is going to clarify a whole lot of things for you as you move through this program. So we try to build it so it's kind of a slower pace in the beginning that you kind of get started, and then it's more and more independent, right? And so that goal for you is possibly to apply some of what we've learned and develop an independent project. Now, another thing that I wanted to mention is that we have social media groups that we invite you to join. I think it's a great place to keep in touch even after you have completed this program. Potentially, we hope that you'll be able to develop some relationships during this time, work on projects together as a group, and so really keeping in touch with others in the program. This is where you can do it. We have a group on LinkedIn. Beepsa, if you could post those links. So we have a group on LinkedIn. We have a group on Facebook. We welcome you to join those and continue interacting there together. So the thing that I asked everyone to take a look at last time are some courses that you can start exploring. So we'll go through some of that content in the next couple of sessions, but also to take a look at some of these publications. So did anyone have a chance to read through the publications that are here on the screen about the Human Microbiome Project. Has anyone had a chance to look at those? All right, ready? Okay, so a couple of you did. That's really good to know. So as you probably discovered, okay, great. So I think this is an interesting introduction into how kind of a little bit of a historical perspective on how these large-scale microbiome data sets have been generated, and kind of what is the history and how did we arrive, why these tools that we're going to be using were developed. So for the rest of you that did not have a chance to look at these, again, the links to some of these publications that we reviewed during the sessions are going to be posted on the forum. You can find them there. Specifically, these two, I'm just going to copy the link for those of you that want to look into this. Please copy, please copy this so that you can take a look at some of these publications. So what are these publications talking about? By 2006, the Human Genome Project was over. It was complete. So multinational undertaking, which began in 1990s, produced sequences of the DNA that make up the human genome. But also, an important outcome of the Human Genome Project was were advances in DNA sequencing technologies and bioinformatics methods. The Human Microbiome Project actually grew out of the Human Genome Project, and it began in 2007. So today we're 2020, it's just 13 years ago, really a recent project. The research was funded by a 153 million grant from multiple national agencies, and the program had several goals in mind. So one, it was to develop a set of reference microbial genome sequences from the human microbiome, and to begin the preliminary characterization of the healthy human microbiome. So one is just a reference data set or database. Two, it's to start exploring the relationship between disease and the changes in the human microbiome, which was an area that was poorly understood. Remember that before that, primarily what people thought of was a particular pathogen, a microbe that caused disease. They did not think about changes in the community in the microbiome associated with different human conditions. Three, it was to develop new tools and technology that could be used to analyze all of the DNA sequences present in the microbiome. It was also to create a place to store and share data with others, create a place to store physical samples of DNA and microorganisms that can be accessed by other researchers, and consider some of the ethical, legal, and social implications of the research. So really the project resulted in a number of important findings. But they started this project by taking, sampling the microbiome across 242 healthy adult volunteers. They took samples from the nose, mouth, stool, skin, and vagina of the volunteers. Scientists resampled the volunteers over the course of 22 months in order to observe how a person's microbiome changes over time. So in total, about 11,000 samples were taken. The sequencing of the human genome was a giant undertaking, but the scientists also had faced other types of challenges, not just the amount of data, but also the fact that the human microbiome consists of thousands of different species of microorganisms, each with their own unique genome. So adding to this fact is that many of these microbes could not be studied for what is their function, what do they do, how do they look. It's very difficult to separate them from the community itself. And so to tackle this, some of these challenges, we will talk about some of the sequencing methods that were developed as a result, and then we'll talk about how those sequencing data sets were analyzed or could be analyzed. Okay, so the other thing that we wanted to talk about today is some of the materials that we also you can already get started with, which is Introduction to Metagenomics and Metagenomics 1. So I have two questions. Let's see what everybody thinks the answer to these questions are. And then we'll talk about some of these topics. Okay, so number one, if you're interested in studying the diversity of bacteria in the feline microbiome, including low-frequency species, which method should be the best choice? So what do you think? Okay, Shreya thinks it's C, Divya thinks it's D, and Dars thinks it's C. Okay, anyone else? Rebecca thinks it's B. Okay, Singh also B, and Dithi B. Okay, so great. So we found a question that we have some doubts about. So okay, Michael and Cam think it's C. Divya also thinks it's C. Okay, so let's take a look at what are these options. So A, shotgun genomic DNA sequencing. B, shotgun transcriptomic RNA sequencing. C, 16S amplicon sequencing, and D, 18S amplicon sequencing. So let's talk about what are these different types of data. We'll talk about this in greater detail, but just to answer this question. So the answer for this is actually, the correct answer is C. And the reason for that is because the 16S amplicon sequencing allows us to study diversity among all or the majority of the microorganisms. So look, we are looking here at diversity of bacteria. That's the goal. So we'll talk about what is 16S amplicon and what is this gene, 16S ribosomal RNA today. But why not some of these others? So 18S, obviously, that's from a different group of species. So this is going to be from mammals, for example. You can look at this 18S ribosomal RNA. But when we talk about bacteria, this is going to be 6S ribosomal RNA. But what about these two? So why not these? So when we talk about genomic DNA sequencing or kind of whole genome metagenomic sequencing, really one of the challenges is having those reference genomes for the different bacteria. So many of the references are not going to help us because of the variation. So the sequencing or the mapping quality that typically we're looking for is not going to allow us to map accurately all of the reads to the references that we have, because we do not have all of the references. So we will end up with a lot of assembly of longer sequences. They're not going to be individual reads, but they're going to be contigs, so collections of reads combined together, and not really a good reference for them. And the same reason is the shotgun transcriptomic RNA sequencing. We could use metagenomic metatranscriptomic sequencing, but the problem with it is that, again, we will have some expression of genes. We don't know what they do. We don't know their function. Really a challenge. Now, the other problem is that we have this phrase low frequency species. So when we look for low frequency species in a sample, this might be represented by just a few members of that particular group. And therefore, we will typically lose a lot of information if we're looking to get the whole genome and also know how that genome is assembled. So really a challenging problem here. Let's look at the second question. Shotgun sequencing of 16S variable domains will provide you with what type of information? A, evolutionary changes due to insertion of viral genomes into these domains. B, a way to infer phylogenies in and among sample specimens. C, correlations between mutations and constant and variable domains over time. And D, ideal locations of amplicons to target for sequencing. So which one do you think it is? All right, excellent. So a lot of you are saying B, and that is correct, because really the 16S ribosomal RNA allows us, it is a well-conserved gene that has similar purpose, so a close evolutionary relationship between multiple bacterial species, and we can study the relationship among the samples and within samples. So let's talk about some of those today. Now, before we continue with kind of today's topic, I wanted to make sure that the questions from last time, so some of the people had some questions about maybe finding the resources, being able to run some analysis with the sample data sets. I just wanted to make sure that all of those questions were addressed, and everybody has completed their profile and started looking at some of the courses. So if you have any questions that you want me to address about some of the resources that we've discussed, please let me know. Okay. Yes, Shreya, excellent question. I am planning to do that today. So today, that will be actually the assignment for you at the end, is to find your own data set on microbiome from NCBI, but we will actually go through that towards the end of this session. Any other questions that you're kind of anticipating maybe, or that you want me to address now? All right, great. So let's move on. So what we want to start talking about today is some of the basic terminology. I know probably most of you are familiar with this, but nevertheless, let's review it together. So what is similarity and homology? So when we speak about relationships and we want to understand how different species or members of those species are related to each other, right? So we identify an organism. The typical approach is to look at similarity. So we want to understand similarity. You can think of that similarity from a function perspective, because how do you know what is closer, a leopard and a tuna, right? How do you actually define that? Well, the approach to measure that distance is to look at how closely related, for example, is a homology between limbs, right? So here you can see the relationship between humans, turtles, bats, and whales, right? It could be studied, or their closeness could be studied by looking at the different elements in the limbs and looking at how close, if they do share the same properties, we can say that they are more closely related, right? So in the same way, we can actually look at gene sequences. For example, we know that certain genes are performing very similar functions. For example, the Hox gene controls body segment development. So these Hox genes, which is a subset of homeobox genes, are a group of related genes that specify regions of the body plan of an embryo along the head-tail axis of animals. So as the embryo develops, those different sections develop at particular times, so they're activated at different times. And not so surprisingly, evolutionary speaking, right, we can think of these genes being conserved, because all different species, from flies to mice to humans, grow from an embryo and develop in a certain defined period of time. That process is conserved, as opposed to, for example, growing hair, right? So growing hair is also a function. It's some kind of a phenotype, but it's not as conserved. And so we can look at genes that have different levels of conservation. They have similarities, and the closer they are, we can start building a relationship between them by thinking of common ancestry. So the way to relate that information is to think of a phylogeny or evolutionary relationships that really show us the time between ancestors and present-day species. If we look at the relationship at the sequence level, we can also measure it by similarity between nucleotide or amino acid similarity, right? So that multiple sequence alignment or similarity of sequences can help us understand the relationship between these different individuals that we observe today. So let's take a look at the tree of life. This is not going to take a long time, But let's take a look at the tree of life. You can find the tree of life, an interactive tree of life, right here on itol. I'm just going to paste it right here, itol.embl.de. So let's take a look at a couple of examples here. Okay, so if you go to itol, and let me kind of zoom out here. Okay, so let me know once you have navigated to itol, this website. Okay, so Cam is there. All right, great. Let's wait for everybody else. So once you navigate to that website, you'll be on the front page, and so you have to go to the tree of life, which is right there. You can also use it to visualize your own phylogenies, but okay, so for those of you that are here, you can see here, I've activated a couple of things that you can add or remove. So for example, you can see genome size. Okay, so just by looking at the genome size, where do you think are the mammalian genomes? Was everyone, by the way, able to load this fully? Or is anyone having trouble finding the site? Okay, so purple. Okay, anyone else? Mammalian, where do you think are the mammalian genomes? Pink, okay. All right, looks like Harpreet found them. They're the pinkish ones. In fact, we have right here, you can see colored ranges, right? So these are the different eukaryota, bacteria, and archaea, right? So these are the three main portions of the tree of life, right? So correctly, you can see that the genome length, you can see over here, genome length is kind of associated also with some of those bigger organisms. So you can scroll through here and see which ones, and right here you have Homo sapiens as well. Okay, so where is the bacteria? All of these, right, all of the purple ones are actually bacteria. So let's take a look at some of this bacteria. Now we can zoom in. Okay, and let's take a look at one particular example, just so that we understand this concept of similarity. So here I've identified this little clade of this phylogenetic tree. So if you can find chlamydia, you'll see a number of different chlamydia members here in this portion of the tree. Okay, so how many of you have found chlamydia? All right, great. So what is interesting about this chlamydia, right? If we look closely at this portion of the tree between chlamydia, you can see that there is this portion of the tree, right? So which actually all of them is chlamydia pneumoniae, right? So all of these, 1, 2, 3, 4, these are all bacteria that cause pneumonia. Okay, so all of these, if you actually look at one of them, you can see maybe Google search or let's see, maybe we can zoom out just a little bit to see. Yep, okay, can't really see it. Okay, so yeah, we can actually look at it from the search, right? And here you will see it's a major cause of pneumonia, and there are a number of different strains. So you can see those strains right here, right? So here you can see those different strains at the end right here. So all of these, and as you look at these other ones, right, they are starting to become different. So as you look at them, they become different, right? So let's take a look at what are they actually. So this one is the most common infectious cause of blindness and the most commonly sexually transmitted bacterium. So you can see that the same member, right, can have many different times, many different functions, but their relationship is in this clade. So that's something that we can use to understand the relationship between these different bacteria. So again, if we're not able to clone and if we're not able to grow this bacteria in a lab, it only some of the members could be studied in detail. Some of the other members could be studied based on the relationship to these known species. So if we look for this particular member, for example, here, you can see the phylogeny is based on 16S ribosomal RNA-based LTP release 123, right? So you can see that the characterization of the relationships between these different members of this clade is actually based on one particular gene. And this is this 16S ribosomal RNA. So what is that 16S ribosomal RNA? Well, we have the process of DNA transcription into RNA and then the translation to protein. And this is performed by the ribosome. The ribosome actually consists of two portions, and it's at the core of this translational machinery for all organisms. And it actually performs two functions: the decoding of genetic information contained in messenger RNA and the formation of peptide bonds. It actually consists of this ribonucleoprotein particle of 70S in prokaryotes, including bacteria, and 80S in eukaryotes, which are composed of two subunits, the 30S and the 50S subunits in prokaryotes. So the smaller one is the 30S, and the 50S is the larger one, and 40S and 60S in eukaryotes. As the ribosome is universal and is submitted to strong selection pressure, ribosomal RNA, or the rRNA, has been extensively used as a reference molecule in phylogeny and is at the origin of the division of living organisms into three domains: bacteria, eukarya, and archaea, right? So when we looked at this tree of life, we are actually looking at how these different organisms are related to each other or different from each other based on their ribosome. So the ribosomal RNA genes, or rRNA genes, are known to vary much less across this tree of life than most other genes. They are actually an important part of understanding of how these bacteria and different organisms reproduce. It is slow to change. It's only 1,500 nucleotides for the 16S ribosomal RNA. And there are multiple different segments in this gene that have a variability. They have different levels of variation across different taxonomic levels. And so what we'll look today is how can they be studied so that we understand what is in our data. So here you can see these two portions, the 30S and the 50S. It is actually a portion that we are sequencing called the 16S portion of the 30S subunit. There's the small subunit and the large subunit. And you can see what size these ribosomes are. So the ribosome itself is actually about 500 nanometers small. So you can see this is 500, yeah, this is the scale, and here you can see it's about 10 nanometers is the ribosome itself. So it's fairly small. So what do we do when we think about how to generate data from the 16S ribosomal RNA gene? So to generate data, there are several different steps to prepare this data and actually do the sequencing. So first of all, the sample is it's extracted from wherever we want to study. For example, from spit, from sputum, or from fecal matter. And then that sample, actually, you want to extract DNA or RNA, right? Depending on what is it that we want to sequence. Whatever that material is, that genetic material, it is then shattered into small pieces, and then it could be sequenced. Now, when we think of sequencing, there's actually multiple things that could be amplified for sequencing to have enough of that genetic material. But as a result, generally speaking, from any type of sequencing, what we get are these short reads. And these short reads could be in different types of format. For example, they could be sequences that we can map to a reference genome, to a known organism, or we can combine them into groups that are similar to each other. And so these are the two main approaches of sequencing, and they will actually have a huge impact on how do we actually look at the data and what we can study with that data. So overall, there are several different approaches to understand the microbiome. We can look at taxonomic profiling, which is to study how they are related. So similar to what we saw in Tree of Life, we can look at the taxonomy tree, and we can look at that taxonomy tree at different levels based on how much information we have for the different levels of taxonomy. Or we can try to understand specific members of that community and try to infer the functionality. So we actually want to sometimes understand what are the specific functions that these genes have, what is the function of those sequences. So when we talk about the 16S ribosomal RNA gene sequencing, it really gives us the ability to describe complex microbial communities without knowing necessarily all of the individual members of that community. So to classify, we can use just this one portion, or even a smaller portion of that gene, as we'll see later. And all that we can get out of that type of data is how are they related within a sample or between samples, to map those relationships and to study what we call species richness. So we can look at what type of species or what type of taxonomic level is present in greater proportion to the overall data that we have. Another approach is called metagenomic sequencing. So when we have this good estimate of who is there, this data does not tell us what could this microbe or what this group of microbes could be doing inside the body. So to get a better idea of what it actually does, you can do whole metagenomic sequencing, which is the total DNA extracted from this community or the environment. And so when you look at those sequences of DNA, one of the challenges is you can map that because we're talking about shotgun sequencing, so we're talking about short reads. And when we look at these short reads, it is challenging to combine them into longer sequences and understand how do they actually map to a reference genome. But it could be done. So if we can do that, we are going to, one, lose some of the low abundant portion of our data. Two, we might not have a good reference. And three, we might actually expect a lot of noise. And this noise part is also related to the type of sequencing that we can do, right? So sequencing could actually be done using multiple machines. The newer ones, MinION, for example, or some of the ones that have been around for a while, like the Illumina. Typically, Illumina MiSeq is used for metagenomic sequencing, but also some of the other methods of sequencing, including PacBio. So does anyone know what might be some benefits or limitations of some of these sequencing methods Has anyone had a chance to work with MinION data? Okay. So the gold standard, okay, great, Michael. All right, excellent. So some of you have that experience. So what is important to understand about the different technologies listed here is the length of the read, right? So the size that they can read, yeah, that's correct. So when we look at these different sequencers, they can produce longer or shorter reads. But also the reliability of those reads, that is another factor that we should consider. So for example, MinION produces fast and cheap longer reads. However, their quality of read could be limited. So there's a lot of errors, a lot of less reliability on each nucleotide that we get out of there. For Illumina, there are multiple approaches like the pair-end sequencing that allows us to get high quality reads, but they're typically going to be shorter. And PacBio is kind of the more expensive, much longer reads and higher quality than MinION. So when we think about different challenges involved in metagenomic sequencing, all of these tools are going to be very useful. So whenever you're looking for that data on NCBI, for example, or in a public domain repository, always ask the question, how was it sequenced? And that will give you some idea into what kind of information you can actually extract from the sample. So when we talk about 16S amplicon sequencing, we are talking about taking that 16S ribosomal RNA gene. So that gene is about 1,500 nucleotides long, or 1.5 kilobases, and it includes a number of different hypervariable regions that could be amplified using specific primers. So here you can see the full 16S ribosomal RNA. The bottom part you can see is the 30S ribosomal subunit. And these are some of the hypervariable regions that are either more variable or more conserved between different species. So here you can see in terms of the mean frequency of most common residue in 50 base window, based on the base position in the 16S ribosomal RNA gene. So here you can see some of those 9 different regions. So when we talk about 16S ribosomal RNA amplicon sequencing, typically we are going to talk about maybe 1, 2, or several of these hypervariable regions. So there's a total of 9 of them. They range between 30 and 100 base pairs long, and are involved in the secondary structure of the small ribosomal subunit. The degree of conservation varies widely between these hypervariable regions, with more conserved regions correlating to higher level taxonomy, and less conserved regions to lower level, such as genus and species. While the entire 16S sequence allows for comparison of all hypervariable regions, it is 1,500 base pairs long. So who knows what is the average length of the Illumina read? Does anyone know what are some of those read lengths? All right, great. Divya, yes, that is right. So 300 is a good kind of an average for the length of the read. So if we have, yeah, or 250, so around that, but if we have 1,500 base pairs, right, what you end up with is that you do not have a full read that can cover the particular, that full sequence. So the limitation of shotgun sequencing is really the length of the read, and the length of the read also limits your ability to reconstruct the full sequence. So the less you know the full sequence, the less confidence you will have that what you have assembled is actually the the correct thing, especially if you don't have a reference. And so as a result of that, the 1500 base pair long sequence becomes prohibitive, but the individual or maybe even a couple of these hypervariable regions together could be primed so that you could amplify them. And for example, here you can see the variable 4 region, so V4. You could just amplify that one, or you can amplify a couple of them. You can see that some of them are longer, some of them are shorter. And as a result, this is called amplicon pooling and sequencing. And typically that is going to be using the MiSeq. So Illumina sequencing typically is 75 to 250 base pairs long, but up to 300 base pairs with Illumina MiSeq. And so that is why MiSeq is going to be typically used for this kind of sequencing. And you have specific primers that are available to amplify that, the different hypervariable regions. Now, as a result, what do you get from the sequencing? You have a number of different formats. So there's 3 different formats that we can talk about in the context of sequencing. The raw output. This is the FASTQ. So FASTQ format is a format that contains individual sequences of reads. Those will have both the read sequence and the quality for each one of your characters, each one of the nucleotides, as well as the length. And so this will be just a collection of those short reads. Then you have a FASTA format. And the FASTA format is typically a format to store longer sequences. This could be storing sequences of whole genes, genomes, or even proteins in amino acid sequences. But typically we would use that for a reference genome. So if you have a reference genome or you have a collection of reference genomes, those could be stored in a FASTA file. And typically also there's a genome transfer format, and that genome transfer format is used to annotate the FASTA file. So to identify specific portions of the sequences that have information about the beginning and the end of some kind of an element, okay? So when you think of that process of generating short reads, ultimately those short reads need to be aligned to something. And so typically what you will get is something that looks like this. So here you have a bunch of individual short reads, and those have to be mapped to some kind of a reference genome. And the abundance of read mapping on a particular position in your genome, when we talk about a genome, or you can think of this as a whole series of genomes, this could be measured as depth of sequencing, which means that you have reliability of that particular position, or even variation in that position. So depending on what kind of sequencing is done. Later on, this could be also annotated based on whatever you have in your reference genome. So when we talk about the FASTA file, which is where you might have your reference genome, it is typically just a sequence of nucleotides, and that could be organized by organism. So here, for example, this is an organism called Spotiptera frugiperta, and this is just a scaffold, so it's just part of the genome. And so you can see the sequence, it just has the individual nucleotides in there. When we talk about the FASTQ file, right, the challenge that we're trying to solve is take each one of our short reads and map it by this short length, 160 or whatever the length sequencing that we have, map it to the reference genome. Now, the intermediate format that shows us how those sequences are mapped onto the reference sequence is typically stored in a SAM or a BAM file. And so it's important to kind of, understand what are these different file formats, because we will then take a look at them and we'll use them in the different types of analyses that we will conduct with these files. So here you can see an example of a BAM file. You can see the BAM file right here. So this is your reference portion, and these are the individual reads. And you can see that it's done by aligning the sequences and introducing gaps wherever those gaps are either present or not present in your reference read. Now, there is a benefit to Illumina pair-end sequencing that gives you higher confidence about the particular position, each nucleotide. And so many times when we talk about Illumina metagenomic MiSeq type of Illumina sequencing, those are going to be paired end. And so that's just an important part to understand. Now, sometimes we can look at the sequences like this, just as letters, but sometimes we can also visualize how those reads map onto the reference genome. So let's do that together. Okay, so let's go to the T-Bioinfo platform. This will allow us to do it together. So this is the link. Did everyone, by the way, get set up with the server and got their login information and everything else? All right, great. So let me know once you are here. Okay, I'll wait until at least 5 people get here so we can start doing this together. All right, great. Okay, so here, what we'll first just explore together is how to visualize alignment of reads to the reference genome. So, you'll kind of understand also the logic of some of these tools. So please go right here to RNA-seq chip and click on this drop-down button. And let's go to this first demo. Okay, so once you click here, you should see something like this. Okay, and please stop me if anyone is having any kind of a technical problem here. What we'll do is we'll take some data, so we won't worry about what data it is right now. But then we want to just take one sample and map it onto the reference genome. So we're going to use a known method called bowtie2G. Probably many of you are familiar with it. Basically what it does is it maps those short reads onto a reference genome. And then after you have clicked there, you should be able to see visualization light up. And so what we'll do is we'll just visualize how the short reads map onto the reference genome. Okay, and at the end, press end. And once it is done, it should reload and you should have this button called visualization. So did everyone get there? Okay, if you have any problems loading this, please let me know. So if you click on this visualization button, this is what you should see. All right, great. So this is called JBrowse. Did everyone get here? So if you got the visualization, please put it in chat that you can see the visualization. So I can see that Kim Abbott can see the visualization. Does anyone else? All right. All right, great. So if you don't and you have some problem with it, just let me know. So what are we looking at here, right? So this is these little, if we zoom in, right here you can zoom in, you will be able to see some of those reads and you can click on one of them and you can see all the information about that read. Now what we are looking at is actually a BAM file. So this BAM file looks at this particular sequence. You can see that this is a sequence of 150 nucleotides long. It has variable quality across the nucleotide sequence, but it is mapped onto a particular reference in the genome. So if we want to see where does it map to, we have to zoom in to see the sequence. So we can zoom in more. Okay, and here we can start seeing the sequence and the reading frames and how they are converting to amino acids. So this is actually the raw data that has been processed, right? We have aligned it to the reference genome. How to select the pipeline again. Okay, so this is what the pipeline should look like. Okay, so you press on start, bow tie 2G, which is mapping on genome, and then visualization, and then end. So Akil, let me know if you've got that. All right. Okay, so what do we see here? We see the individual reads. We also see some variation. So here you can see, for example, a G as opposed to whatever we have in the reference genome, right? So this is a difference. And sometimes that difference is going to be consistent across many different reads. And that will be like a variant in those reads, right? So when we look at this type of an alignment, this is alignment to one reference genome, and the information that we can have are the variations from the reference or abundance, right? So that's another thing that we can get out of this, is how abundant is a particular region. So for example, this shows us individual reads. You can see here, this is the alignment, or we can just look at abundance. So this is coverage and SNPs. So here you can see only certain regions are covered, right? Typically, when you look at this kind of an alignment, you also will have multiple portions to select from. So these could be chromosomes. Again, where did these separations come from? These are, if you think of a FASTA file, they will have that reference written out there. If you have a human genome, for example, there will be chromosomes. If you have a database of several FASTA files combined together, for each one of them, you will see that right here. Okay, so did everyone have a chance to explore this kind of visualization? Please let me know. And what we'll do now is we'll try to apply this to 16S ribosomal RNA data. Okay, so did anyone have any questions about this? What we're seeing here? What are the FASTA files? What are the FASTQ files? What are we doing by aligning those reads to the reference genome? Okay, any questions? Okay, so the color coding is, if we open the reads, there are two colors, blue and red. These are the forward and the reverse. So this is pair and sequences of reads. So one is red, one is blue. Okay, and typically I think you can, okay. How can we know which pipeline to choose? Great. So now this is just a simple alignment to the reference genome, right? But we're talking about, when we talk about the microbiome sample, we're talking about a group of reference genomes, right? So how do we actually do that? Let's go back to the main page. So you just go to areas of analysis. And then you scroll down. Okay, and we'll talk a little bit more about this in our next session as well, using some examples. But we'll move step by step. And so you can see now that instead of reads, and I'm just going to close this, instead of reads, you can actually open this section right here and click on Metagenomics regular. Okay, so please click here. Let me know once you're here in the metagenomics microbiome. Okay, so put in yes or I'm here or something like this when you get here. Okay. Just typically what we'll do is we'll wait until at least 5 people get to the same page so we don't lose everyone. All right, great. All right, so again, we'll press on start. And now you can see that we use the same method, right? Bow tie 2. But instead of mapping on a genome, we're going to map onto the reference database. So here you can see that there are databases like Silva, for example, that contain many different 16S genes from many different organisms. So you click on OK. And then once you map them, instead of looking for variants or instead of looking for specific level of something, you're actually looking at abundance, right? So this is just looking at how many reads from a sample fall onto a 16S gene from a particular species or a group of organisms, okay? And once we do that, once we calculate how many, we also want to use the information in this reference database that tells us where did that gene come from. So what type of bacteria did it come from? Okay, so names replace and then end. Oh, okay, Kim, we're not actually running the analysis, we're actually using the demo. So when you are on here, don't use this one, use the demo. Okay, we'll get to actually run this in the future, but right now we just want to do a demo quickly together. Okay, so names replace and then end. Again, it will reload and you should have a number of different output files here. So let me know once you've got those output files. Okay. Let's wait until at least 5 people get those output files. All right, 4. One more. All right, great. Excellent. Okay, if you are having a problem getting to these output files, let me know. We can try to address it. But here you will have a number of different outputs. We have metagenomics joint table post count, mapping stats, bow tie annotation, and table after annotation. So let's take a look at the table after annotation. Okay, this is going to be a TXT file, but it's actually a tab delimited, so you can open it in something like Excel, or you can open it in R, or whatever you want. And let's just take a look at this very simple table. Okay, I'm saying this is a very simple table because it actually contains the annotation for the different 16S RNA genes. You can see what level of taxonomy each one of these came from. And you can also see that this is result for just one sample, ERR 1250035. Okay, so let's take a look at what is this file. Let's go to NCBI. Okay, and you can see that this is a sample from the Human Gut Metagenome, the American Gut Project. Okay, so you can see that a little information about the American gut, and all of this information is inside Metagenomics 1, right? So if you want to actually run this analysis yourself, you will be able to find these data sets and also learn a little bit about the Human Gut Microbiome Project in that sample, in that lesson, of course, sorry. Okay, so what is this? This is actually the number of abundance, so to say, for this particular sample. So if we sort this, you can start seeing abundance of particular species within this sample, right? So for example, this one, I want to understand what this is. You can see that this is a genus of bacteria that are gram-positive and aerobic. They're bacilli, and in some phase of life they are. Okay, so you can read about this particular bacteria. What does this tell us about this sample, right? So it's a question of what it could actually tell us, but the interpretation, right? The interpretation, the very immediate, simple interpretation from this is that in this particular sample, and we don't know right now anything about the sample, who did it come from, or what is the phenotype associated with this sample, but in a very basic way, we can see that there are several members that are highly enriched compared to other ones. Okay, what other things can we see? For example, if you look through, right, you look through here, you can see that all of them belong to bacteria. Some of them are actinobacteria, some of them are proteobacteria, some of them are firmicutes, right, fusobacteria, so that there are different levels of taxonomy that we can look at to understand the abundance, because this abundance is for the lowest level of taxonomy. If we go to the higher level of taxonomy, we could separate them by the different categories, and we can learn other things about the composition of this particular sample. Okay, so does that make sense? In a, I know that this is still kind of very basic, but the idea, I think, should be clear from this one single example. Okay, did anyone have any questions about this? Okay, so now we know that, okay, what's the difference between row 2 and 3? So let's take a look. So you can see that the ID is different. So if you want to understand more specifically, and you are correct that this is something that with this table, you're going to start working with that table. So this is from the Silva database. Hold on. Okay, so here you can see that this is an accession number. Okay, so this is going to be present also on NCBI. So let's take a look on NCBI. Okay. So this is probably going to be on nucleotide. So let's take a look. 403. Anyway, so this is going to be on NCBI or the Silva database, is where you will be able to find the specific ID, right? And that's what is different here are the different IDs, right? And then this is the level of annotation that is known. For example, if they are exactly the same, maybe they are different variations within this Carinobacterium. Okay, so you can see species for both of these is not known. So again, the level of taxonomy, the level of annotation for all of these is not always detailed and known. And so that's why we basically group by different levels of taxonomy to really study the variation. So Rebecca, if you want the specifics of what this ID does, I'll actually take a look at that later and get back to you. But other than that, the concept, you understand the concept, right, Rebecca? All right, great. So there you go. There's histamine receptor. Okay, I'm not sure that's that because it's actually the 16S ribosomal RNA gene, right? So I'll look for it later and I'll kind of update. But the idea is that you have an ID. This is an ID from the Silva database, and that is annotated by different level of taxonomy. And based on what level of taxonomy you want to use for your composition, that is the information that you can get out of this table. Okay, so what did we learn? We learned about different types of data formats, and we understand what information is within each one of our data types or data samples. In this case, we looked at a single sample, we looked at how the reads are mapped to a reference, and then we took a reference, and there are multiple types of reference databases that we can choose from. For example, we can use the database on NCBI, or Green Genes, RDP, and Silva. And Silva specifically has been looked at in terms of its reliability and curation and active participation of a community. So it is typically used for this kind of 16S ribosomal RNA type annotation. All right, so about this process of sequencing, generating data, and then mapping and annotation on a reference, in this case, a reference database, are there any questions? Okay, so there are no more questions. What I suggest is going through this American Gut project, right? So we already looked at one example. So in this data set, we looked at this example. This data set, by the way, moves around. And so the latest project consolidation that happened is there's a new site called microseta.ucsd.edu. You can take a look at that. This is an initiative that consolidates the global efforts to collect microbiome samples of the world's population and to couple these collections with educational outreach about microbiome science. So you can see that TMI is a part of the Earth Microbiome Project, and they have a number of different sub-projects, and American Gut now becomes a part of this initiative. So the American Gut Project was really started as a way to study self-reported data sets. And what you can get out of that in this type of an analysis is actually looking at how different types of microbiota could be compared between an individual and the other participants of the program. Now, in detail, this information is going to be reviewed in Metagenomics 1. So you'll actually learn about the project, learn about the different methods of sequencing, and then try that analysis using different types of attributes like geolocation and age and other types of things that you'll be able to find about that project sample that we did. Now, importantly, when we do this kind of an analysis, you have to remember that we are doing a fairly simple analysis. We're just taking the short reads that we have from the input data. Using a reference database, we align them to this reference database. And then we count how many, just a very basic count, how many reads do we have in each one of our positions, and then add the annotation at the end. So the analysis is very simple. As a result, you have this type of an output, and this type of an output really shows you different levels of taxonomy that now you can compare within a sample or between samples. So between samples will give you an idea of how are these samples compared to each other. As a result, based on different levels of taxonomy, these are some of the findings. So some of the findings could be that in different regions of the body, the oral cavity, stomach, colon, vagina, skin, etc., you have different abundances of different species. So here, for example, firmicutes are blue, right? And you can see that they're abundant in the vagina, not so much in the colon, not so much in the skin, for example. Or if you look at the actinobacteria, right, it is very present in the skin, also in the stomach, for example, in the colon and the vagina, almost not at all, right? So when we look at this table, right, we see the actinobacteria. So we really need to know what is the sample of to understand whether what we are seeing is normal or expected, or is this some kind of a dysbiosis? And in our next session, by the way, we will talk about what are some of the impacts of this kind of compositional studies on the functionality of this bacteria, on how do we evaluate whether it's normal, healthy, or there's a dysbiosis that we can infer from what we understand. Okay, so after this, I just want to show you how to find some of this kind of data on NCBI. But before we do this, are there any questions? All right, great. So if there are no questions, let's all go to Easy BioCloud. Okay, so let's leave this to the end so that we can actually take a look at some of these data sets. All right, so please go with me to NCBI. Dysbiosis, great. So dysbiosis is when typical composition that is stable is disrupted by some disbalance that is naturally expected. We'll look at that in our next lecture, but please go with me to NCBI. Let me know once you are there. Okay, 2 more people, and all right, great. So 5 people, let's move on. So how do we find such data sets on NCBI? Here you can search between several different databases. So here you can see all of these different databases. And the first thing that you can do is obviously just type in Metagenomics. So if you type in Metagenomics and search in all of your databases, you will get results for all the different databases that are available. What are some of the databases where you would expect to find data that we could actually analyze? So we'll take a look at some of these different databases, and then we'll see what kind of data do they contain. So the first one is the bioproject. Okay, bioproject is a collection of different types of studies. So think of them as a publication that collected many different samples. They are organized in bioprojects, and inside they might include a number of different types of data. For example, metagenomics analysis of the gut of Eri silkworm larva, Samia racini, or intestinal microbiome and complement system in age-related macular degeneration. Okay, as you look through, you can look for some more interesting studies to you. For example, characterization of the preterm infant reservoir of antibiotic resistance genes, or etc. Sewage metagenome. So whatever is interesting to you, right? If you take any one of these, inside, you will typically have all the different types of data that are available. For example, the next example is SRA, right? So when you actually, you can read here about what they did. For example, here, we used 16S ribosomal RNA amplicon sequencing to analyze the natural microbiome present in the water treatment plants in 2 rural communities and 1 city of the highlands, etc., right? So here, you will find the actual data. If you click on the SRA experiments, you can see for each one of your samples, you have a separate entry. Okay, here you can go to send to and generate a file, or you can see view results as an extended interactive table using the run selector. So send results to run selector. Let's see what are these samples, right? That will give you some metadata about these samples. So here in the metadata, this is right here, you can see isolation source. And this is important because when you think of a study, you're obviously interested in how it was sequenced. So how was this sequenced? What was the method of sequencing for this particular data set? Does anyone know what was the method of sequencing? Okay, the method of sequencing was 16S ribosomal RNA, yes, with Illumina, but it was 16S amplicon data. How did I get here? Okay, so here, did you get to the list of SRA files? Did you get to here? Okay. So when you go to NCBI, right? So, you know, what we did was on NCBI, we looked for just metagenomics or something like that, right? So we searched for metagenomics. And, okay, so did you get to here to just metagenomics on NCBI? Okay, yeah. Well, no, you don't have to go to Bioproject. I just went to Bioproject. Okay, so I went to Bioproject and searched for metagenomics. Okay, then you have a list of projects, and I just picked this one. Well, you can pick anyone. Okay, inside, for example, this has no public data, so this is obviously a bad bioproject. Here's a good bioproject that has some data. Okay, now what I did next is you have SRA experiments. SRA stands for Sequence Read Archive. So these are the raw FASTQ files. So if you click on this number 14, you will see the list of all of the files. Did you get here, Cam? All right. And then here, what I did was send results to run selector. So this is a good way to look at a table that shows you isolation source, location, right? So these are now phenotypes. So we have the raw data, the sequences, and now we have the phenotypes. What are we associating them with? So isolation source, we have location, we have province, and then we also have type. So you can see school, reservoir, school, whatever. So all of this information is going to be important for you to do the analysis later on, because the analysis will consist of finding association between something in your data and what you know about the data. Okay, so let's take a look at a few other examples. Here's an example of a bioproject. Okay, so here you can see understanding the biological factors underpinning the structure of the nasal microbiota. So approximately 20% of humans persistently carry Staphylococcus aureus in their nose. Evidence suggests that the basis of persistent carriage includes a host genetic component and that the microbiota is different in carriers and non-carriers. The biological basis and interaction between the two states is unclear, and there are two alternative explanations. Host genetics may determine S areas carriage by enabling S areas to colonize the nares, which then in turn causes a shift in the constituents of the nasal microbiota, right? So again, your question was about the dysbiosis. Well, dysbiosis is a change in the composition, and that change could be driven by a particular member that gets out of control, right? Causes dysbiosis. Or B, host genetics may determine the constituents of the nasal microbiota, which then either prevent or promote S area survival or persistence. The study aims to investigate the link between host genetic nasal microbiota structure. Okay, how many samples do we have here? $859, correct. So here you can see S array experiments, 859. If you click on 859, you will see all of them. And if you want to understand which one came from where, right? So obviously, if we're studying a nasal microbiome, it came from individuals, came from patients. We need to know something about them. Some of them are carriers, some of them are non-carriers, right? So we go to send results to run selector. Here we have the metadata. The metadata tells us where they came from. What type of information do we have? We have all kinds of code. So sometimes it's not easy to find what exactly is available. You probably can just click on metadata to learn more about what is available for each one of your tables. So here you can see, just by looking at this, you can see it's CSV file instead of a TXT, which is what it tells me to download. Okay, so I'm just going to change this to CSV. And now it's a table. So what is in here? You see it's metagenomic sequencing. Okay, and here you have some title. So the title is probably what you want to look for to understand what is in the data. Okay, but again, now you're on a path to combine all of this annotation about the samples to plan your analysis. Okay, so when you want to apply some of these methods, you really need to plan it out to understand what kind of information is available. Here is another a data set. This is metagenomics of human microbiome, contribution of the vaginal microbiome, metabolome, and host response to increased risk of preterm birth in an ethnically diverse UK cohort. Okay, what is this? What are we looking at? This is an SRA collection. Okay, so this is actually one sample from a larger, if you see here, it will tell you all experiments or all runs. You can go and find all of the data from here. And here you can see that this is 16S ribosomal RNA. Okay, and again, like we did before, you can go in here and understand what is the associated data. Here's another example. Again, SRA. But remember, when you look for metagenomics, sometimes you also find other types of data. So let's search again for metagenomics. Okay, here's another example. So we looked at bioproject, we looked at SRA. Let's look at GEO. So who knows what GEO stands for? Does anyone know what GEO stands for? Okay, GEO stands for Gene Expression Omnibus. So this is going to be Gene Expression Omnibus. How do you think we can study gene expression from metagenomic sequencing? What do you expect here? So let's take a look. Here's a data set please take a look at this example. We'll just quickly look at what kind of data is available for gene expression of metagenomic sequencing. Okay, what does it say? Microbial functional genes are fundamentally different from bacterial phylogenetic distributions in Arctic soils. Okay, so this is an interesting point to make. We can study diversity and species richness, but ultimately that does not give us too much information about the function, about the activity. And so here they were trying to compare activity and phylogenetic analysis. And what kind of data do they provide? So here you can see all of the different 72 samples. You can download them individually, or please download this data set at the bottom. So this is GSE 63927 raw values. Okay, you can click on FTP or HTTP and you will download that sample. So let me know once you've downloaded that sample. As we're running out of time,